a problem. Can you give some examples when this immunity is a problem?
A transplant.
Very good, excellent. When you have transplanted some tissue, okay, from other person, that is graft, then you want to suppress the immunity. So in organ transplant, yes. Any other reason why you want to suppress the immunity?
Autoimmune disorders.
Excellent. In autoimmune disorders, can you give some examples of
SLE,
SLE? Excellent.
rheumatoid arthritis,
Rheumatoid arthritis, very good. ITP, okay. Any other? Okay, so there are so many autoimmune disorders. Okay, so what is immunity? Immunology, it is the physiology of discrimination of the self from non-self antigens. Okay, so what are the non-self antigens? In the body, the infectious organisms like bacteria, viruses, fungus. Okay, then the second example which you give, that is a graft or a transplant. Okay, and also the cancer cells. Okay, so the cancer cells, they are the self antigens only. Okay, now in Latin, immunis means exempt, referring to protection against the foreign agents. Now, immunopharmacology is the study of drugs which modify the immune system. Okay, either the immune suppressants or immune stimulants. And immunotherapy is the use of the clinical application of these drugs is known as immunotherapy. Okay, so in immunopharmacology, we will discuss both the types of drugs, that is immunosuppressants as well as immune stimulants. Now, can you tell me what are the types of this immunity?
Cell-mediated and humoral.
Cell-mediated and humoral, very good. But before that, what is the basic classification
Innate
of
and acquired.
innate and adaptive.
Very good, excellent. That is innate and acquired. Okay. What is the difference between innate immunity and acquired immunity? Anyone from the class can tell any one difference between innate immunity? It is in its name itself.
By birth.
Innate is by birth. Excellent.
Innate is present since birth and
From
acquired
the
after
Acquired, it is acquired, obviously.
acquiring.
Yes. So innate is
acquired during the lifetime.
present since the birth, and acquired is acquired whenever there is infection, right? Innate is non-specific, okay? Whereas acquired is
Specific.
specific. So we will see what are the types of immune responses in our body. Innate immune response and adaptive immune response. So innate immune response is present since birth. It is non-specific, whereas adaptive immune response is pathogen and antigen specific. Okay. Innate immune response, it is immediate. That is, whenever there is infection, then there is immediate response, that is innate immune response. And it is by a limited set of receptors or components. Okay, limited set of receptors or components. Whereas adaptive immune response, it takes time, that is there is a lag time, and it is antigen specific. In innate immune response, there is no immunological memory. That is, every time there is an infection, right? The same by any pathogen, there is same type of response. Whereas adaptive immune response, there is immunological memory, okay, and next time whenever there is infection, there is a quick response because of this immunological memory. And innate immune response is found in nearly all the forms of life, whereas adaptive immune response is present only in jawed vertebrates. So what are the various types of blood cells? You know, you have erythrocytes, right? You have platelets or thrombocytes, and then you have immune cells like lymphocytes, okay, and you have WBCs, which are granulocytes and agranulocytes. Granulocytes, what are the examples of granulocytes? What are the examples of granulocytes? Yes, basophils,
Neutrophil and basophils.
eosinophils, and neutrophils. Yes, these are the examples of granulocytes. And agranulocytes, the examples are monocytes and macrophages. Okay. So how these cells, they are developed in our immune system? All of these cells, they originate from the pluripotent stem cell. Okay. This pluripotent stem cells, it gives rise to the various progenitor cells like lymphoid stem cells, okay, erythroblasts, megakaryocytes, then monocytes, and trilineage myeloid stem cells. Okay, so lymphoid stem cells, they give rise to T lymphocyte, B lymphocyte, and natural killer lymphocyte or natural killer cells. T lymphocytes and B lymphocytes, they play an important role in adaptive immunity, whereas natural killer cell, it plays an important role in innate immunity. Okay, the T lymphocyte in the thymus, it migrates into the thymus in the presence of thymic hormones, and later on this T lymphocyte is differentiated into various types of T lymphocyte. That is, what are the various types of T cells or T lymphocytes? Example,
T
Cytotoxic,
helper,
very good.
cytotoxic helper cells,
Cytotoxic
suppressor cell.
and T helper cells. Okay, so cytotoxic T cells, they are directly responsible for killing of the organism, whereas T helper cells, they help in the cell-mediated as well as humoral immunity. The T helper cells can be of three types: T helper 1, T helper 2, and T helper 3. Differentiated based upon the cytokines they release. T helper 1 cells, they release interleukin 2 and interferon gamma. Okay. T helper 2 cells, they release interleukin 4, interleukin 5, interleukin 10, and 13. Whereas T helper 3 cells, they release immunoglobulin A. Okay. So we will later on see what are the functions of this T helper 1 and T helper 2 cells, right? Natural killer cells, they play an important role in innate immunity. And B lymphocytes, what is their role?
plasma cells.
B
Antibody.
lymphocytes, they are
I
differentiated into the plasma cells, and these plasma cells, they are responsible for
immunoglobulin production.
production of immunoglobulins, right? That is known as humoral immunity or antibody mediated immunity. Okay. So these are the lymphoid stem cells. Monocytes, they give rise to the macrophage, and granulocytes, they are basophils, eosinophils, and neutrophils. Now, innate immune response. Okay, innate immune response, it is present since birth, and uh, it is mediated by a limited number of components or receptors. It is immediate response. The functions of innate immune cells. Innate immune cells, the main function is phagocytosis. Okay, phagocytosis to kill the offending organism. This phagocytosis may be opsonin dependent or opsonin independent. Then, second important function is proteolytic digestion and presentation of the processed pathogen fragments. Okay, the peptides of this pathogen fragment, they combine with this MHC2 protein on the surface of antigen processing cell. Okay, antigen processing cell, which are mainly the macrophages or dendritic cells or B lymphocytes. Yes, uh, and presents this peptide of the pathogen to the T cells, and this leads to the activation of T cells in the acquired immune response. So, okay, so there is a link between innate immune response and acquired immune response. The innate immune cells, they cause the proteolytic digestion, the peptide pathogen fragments, which combine with MHC2 protein. They are presented on the antigen processing cells, and they activate the T cells of the acquired immune response. And they also secrete the cytokines, which increase both these functions, that is phagocytosis, as well as the second function, that is proteolytic digestion of the processed pathogen fragments. So phagocytosis is one of the main mechanisms of innate immune response, and it may be opsonin dependent and opsonin independent. What is meaning of this opsonin? It is an agent. Okay, opsonin is an agent which coats the invading organisms so that they are better recognized by the immune cells and killed. So what are these opsonins? Opsonins, they may be immunoglobulin like immunoglobulin G or E, or a part of complement, that is C3b, or lectin. So these are the opsonins, and they may coat the invading organisms, which are then easily recognized by the immune cells and killed. Now, the opsonin independent mechanism is mainly because of the pattern recognition receptors. Okay, this pattern recognition receptors, they are belonging to the family of toll-like receptors. Okay, they are also known as PAMP. Okay, pathogen associated molecular pattern receptors. So this pattern recognition receptors, they recognize the generic pattern related to the bacteria and virus. And because of this recognition, for example, the bacterial cell wall components may be recognized by the toll receptors, or the viral DNA components may be recognized by these receptors, or the lipopolysaccharides present in the gram-negative bacteria may be recognized by these receptors, like the toll receptor 4. Okay, even the nanograms of this components, they may be recognized, and teichoic acid of gram-positive bacteria is recognized. So there are at least 10 of these toll-like receptors which recognize the various components of the bacteria and differentiate it from the human cells. Okay. So this is opsonin independent phagocytosis. What are the phagocytotic cells? Macrophages and neutrophils. So in opsonin dependent mechanism, they will be coated by opsonin and then phagocytosed by neutrophil or macrophage. Opsonin independent mechanism, because of the presence of toll receptors, which recognize this components, they will recognize this and phagocytize the invading organism. Now, what is the role of complement system in innate immune response? Okay, so basically the first half of this lecture is the introduction to immunology, which is partly related to pharmacology, more of your microbiology, right? And the second part will be about drugs, so that you understand how the drugs they act in a better way. So complement system is a group of proteins, right? There are at least 20 proteins with complement system C1 to C9. There are 11 such complement proteins with 3 added subunits in this 1 to 9. And in our body, they are in inactive form and they can be activated. The complement system can be activated by a direct pathway or a indirect pathway. Okay, direct pathway is by antigen antibody reaction. Antigen antibody reaction can lead to complement activation. Also indirect pathway because of the uh, because of the lipopolysaccharides, right? And this complement stimulation, it will lead to increased vascular permeability. Okay, increased vascular permeability is by the complement C3A and C5A. Also C4A plays a role in this, and this will lead to the increased entry of the immune cells at the site of infection. Okay, and the second thing is chemotaxis. There will be also increased chemotaxis. Increased chemotaxis is because of the complement C3A and C5A. The complement C5, it breaks into complement C5A and C5B. Complement C5A is responsible for chemotaxis, and complement C5B, it is responsible for the formation of the membrane attack complex, okay, along with the other complements C6, C7, C8 and C9, which cause the bacterial cell destruction. Opsonization is the function of complement C3b. Okay, so the complement system, it plays a very important role in innate immune response by increasing the vascular permeability, by chemotaxis, by also opsonization, and then by formation of a membrane attack complex, which causes this the bacterial cell destruction by cytolysis. Okay, this drug eculizumab, it is a blocker of C5. It prevents the cleavage of C5 into C5A and C5B. Okay, so it will decrease both the formation of membrane attack complex as well as chemotaxis. Now, what is the role of other components in innate immune response? I think this is just a revision for you. Neutrophils, they are responsible for phagocytosis. They also release the enzymes, destructive enzymes, myeloperoxidase and defensins, and eosinophils, mainly the defense against parasites which cannot be engulfed by the neutrophils or macrophages. So this eosinophils, they can attach to the parasite, and then they secrete these cytolytic substances. And also they play an important role in allergic responses. Basophils and mast cells play an important role in IgE mediated anaphylaxis. The IgE antibodies, they can attach and sit on the basophils and mast cells, and whenever there is the circulating IgE antigen, this will attach to this IgE antibody, and there will be degranulation of the mast cells, leading to release of histamine and anaphylaxis. Macrophages and dendritic cells, they play a role as antigen presenting cells. Okay, and natural killer cells, they have the intrinsic ability to kill the invading microbes and malignant cells. There are two mechanisms by which the natural killer cells they play a role. One is by the Fc receptor. Okay, they have this Fc receptor on their surface which recognizes the Fc of the antibodies and leads to the phagocytosis. Okay, and the second is by the killer activated receptors. Natural killer cells, they have presence of killer activated receptors which when activated kill the invading organisms. Now, adaptive immune response, mainly it may be because of cellular immunity or because of cell-mediated immunity or antigen-mediated immunity. So as you have said that the cell immunity, it is by T lymphocytes, CD8 and CD4. That is cluster differentiation factor 8. If it is present on the cell, it is CD8 positive. And if cluster differentiation 4 is present on CD4 cell, then it is CD4 cell. Okay. So CD8 cell directly attack the invading organism, whereas CD4 cells, they aid in cell-mediated immunity, that is T helper 1 cells, and T helper 2 cells, they aid in antibody-mediated immunity. Okay. Antigen-mediated immunity, it is by the B lymphocytes, okay, which differentiate into plasma cells and which synthesize this immunoglobulins. So antigen-mediated immunity is mainly useful against the antigens which are dissolved in the body fluids and extracellular pathogens which cannot go inside the cell, whereas cell-mediated immunity is mainly against the intracellular pathogens. Okay, like tuberculosis, you have seen, like leprosy, okay, or the organisms which can go inside the cells, also the cancer cells and tissue transplants. The main role is played by the cellular immunity. Now MHC complex, I think you know about this MHC complex. What is MHC complex? Major histocompatibility complex. It is also known as human. Yes, I think in microbiology, autoimmunity, this immunity has been covered.
Yes, sir.
Okay, so
Human leukocyte antigen.
Human leukocyte antigen. Okay, because they were first identified on the leukocytes. So these MHC antigens, they are present on the plasma membrane of almost all the body cells, and their function is to recognize the foreign antigen and to differentiate between the self and non-self antigen. So two types of MHC they are present, that is MHC1 and MHC2. MHC1 is present on all the nucleated human cells, whereas MHC2 is present on special antigen presenting cells, that is B cells, macrophages and dendritic cells. What is role of MHC1? MHC1 recognizes the self proteins which are to be ignored. Okay, so it recognizes the self antigens. Actually, it recognizes the blueprint of the self proteins or self antigens which are presented to it from all the nucleated cells. And also, if there is a invading pathogen, okay, inside the cell, that also can be recognized along with these self antigen. MHC2, which are present on the B cells, macrophages and dendritic cells, they identify and display the peptide fragments of an exogenous infectious agents. Okay, these cells, they will dissolve these peptide fragments and they will present it to MHC2, and then this complex of the peptide fragment with MHC2 will present to the T cells, right? And then the T cells will be activated. So MHC1 present on all nucleated human cells, whereas MHC2 limited only to B cells, macrophages, and dendritic cells. Now we come to the important thing, that is the cell-mediated immunity involving T helper mediated activation of T lymphocytes. Okay. So the peptide fragment of the pathogen along with MHC2 in the antigen presenting cell is presented to the T helper cell. Okay. The T helper cell has the presence of this important component known as T cell receptor. Okay. So this MHC2 with the peptide fragment attaches to the T receptor, T cell receptor, and what will happen when it presents to the T cell receptor? Yes, any answers? What will happen?
Interleukin 4 is released.
Interleukin 4 is released.
Toll-like receptor.
Okay. Remember, here just the attachment of this MHC2 or peptide with T cell receptor is not enough. Okay. There needs to be co-stimulation. Okay. There should be co-stimulation. Without co-stimulation, there will be no response. Okay. This is known as T cell energy or unresponsiveness of T cell. T cell will respond only when there are when there is attachment to the T cell receptor, and second when there is co-stimulation. This co-stimulation is by the B7 with CD28. When this B7, that is CD80 or 86, it attaches to the CD28, then there will be co-stimulation, and this will lead to the activation of CD3. Okay, the CD3 on the T helper cell, it will be activated, and the signals, they will be transduced to the nucleus, and that will lead to the transcription of the genes which are responsible for the synthesis of cytokines. Okay, cytokines like interferon gamma and interferon 12, which stimulate the T helper 1 cell, and interleukin 4, which stimulate the T helper 2 cell. Okay, so co-stimulation is very important. If there is no co-stimulation, that is by attachment of CD80 or 86 with CD28, or attachment of CD40 with CD40 ligand of the T cell, then this response will die, and the antigen, there will be no activation of the T cell. This is known as T cell energy. This is also known as T cell energy, or this is the concept of tolerance. Okay, this CTLA4, this ligand, when it sends signal to this B7, there is negative feedback. Okay, that is the immune stimulation is by attaching with CD28, and negative feedback is by CTLA4, which attaches to this B7. So the final thing is release of the cytokines like interferon gamma and interleukin 12, which stimulates the T helper 1 cell, and this T helper 1 cell, it causes release of interleukin 2, interferon gamma, and TNF alpha, again which activates the cytotoxic T cells, and these cytotoxic T cells again they release interleukin 2, and interleukin 2 again activates the cytotoxic T cells. Interleukin 4 is responsible for humoral immunity. By acting on T helper 2 cells, it causes the release of the interleukin 4, 5, 10, and 13 from the T helper 2 cell, which plays a role in humoral immunity. T helper 1 cell plays a role in cell mediated immunity. So this was the T helper cell. Okay. Now, role of the cytotoxic CD8 cell, it is similar. Okay, and it plays a role in cell mediated immunity. The virus infected cell, it presents the viral peptide to the T cell receptor, right? But there will be no response if there is no attachment of this B7 to CD28. If there is no positive response from this B7 to CD28, again this impulse will die and there will be no activation of the T cell. Okay. So this is how the T cell is activated. Also, there is a this FAS ligand. This FAS ligand attaches to the FAS receptor on the virus infected cell, and it leads to the death of the virus infected cell.
Apoptosis.
Yes, by apoptosis. Now coming to the B cell and humoral immunity, whenever the antigen is presented to the B cell, the antigen combines with the B cell receptor, and again this MHC2 complex with the peptide of the pathogen, it is presented to the T helper cell, and when there is activation of this T helper cell with the help of this T cell receptor and also CD28, there will be release of interleukin 2. Okay, this interleukin 2, it acts on the interleukin 2 receptor. The main component of this interleukin 2 receptor is CD25, and this again leads to the T cell proliferation and activation. Okay, and also this T helper cell will release the variety of other interleukins like interleukin 4, interleukin 5, interleukin 10, and interleukin 13, which will lead to the proliferation of the B cells and differentiation. It will lead to the formation of memory cells, okay, which are preserved and are for the next infection. Whereas the B cells, they proliferate into the plasma cells, which cause the production of the antibodies. The first antibody to be formed is, what is the first antibody which is formed? Yes, anyone? IgM.
Antibody.
Which antibody is formed first? IgM. Okay. And because of the attachment of this CD40 to CD40 ligand, there is induction of the protein kinase. Okay. And this protein kinase then causes the switch of the production from IgM to the other antibodies like IgG, IgE, and IgD. Okay, but the first antibody which is formed is IgM. These are the functions of various antibodies which you can see later on. Okay, they are given in Sharma. IgG is the most abundant. IgA 15% present in the mucous membrane. IgM and IgD, they are the antigen receptors present on the B cells, and IgE, it is present on the mast cell. Structure of the antibody. So antibody, it is a Y-shaped globulin. It has two heavy chains, right? This is the heavy chain, and two light chains. The heavy chains, they have around 500 amino acids, whereas the light chain has 220 amino acids. In the heavy chain as well as light chain, there is a variable region. Okay, so this is the variable region. Okay, and the rest is the constant region. So variable region of this antibodies, it is responsible for the antigen binding. It is also known as the antigen binding site. Okay, FAB is again this region up to the variable region, and FC is uh, FC portion of the antibody is this one. So what is the fate of antigen antibody complex? So this is the antigen binding site and the complementary determining region of the antibody, okay, mainly in the variable region. So it has two portions, the FAB region and the FC region. FAB region, it is responsible for the binding of the antigen, whereas FC region, it is recognized by the certain receptors present on the macrophages, okay, and the neutrophils. So whenever the antigen binds to the antibody, there may be opening up of this FC region, and which is recognized by the FC receptor of the macrophages, leading to the opsonization. Or the second thing which may occur is when the antigen attaches to the antibody, or the epitope attaches to the antibody, there is activation of the complement. Because of the activation of the various complements, that is C5, 6, 7, 8, and 9, there is formation of a membrane attack complex, and this leads to the bacterial cell lysis. Okay, so this is the fate of antigen antibody complex. This is the simplified representation of the immune responses. MHC class 1, okay, present on the nucleated cells of the body, okay, and along with the antigen presenting cell, it presents to the CD8 cell, the T cell receptor of the CD8 cell, and also the CD28 co-stimulation. It leads to the activation of CD8 cytotoxic cell, and this leads to the proliferation and differentiation of the CD8 cells, okay, which are cytolytic, and they can directly kill the invading organism. CD8 cell can also be activated by the help of T helper cell, because of the cytokines interferon gamma and interleukin interleukin 2. Okay, so this MHC class 2 antigens, they present the along with the antigen presenting cell, they present the peptide to the CD4 T helper cell, right? And again, the stimulation of CD28 co-stimulation and the T cell receptor stimulation will activate the CD3, which will lead to the release of the cytokines, which are responsible for cell-mediated immunity as well as humoral immunity, and also responsible for the proliferation of CD4 cells themselves, okay, which play a role in delayed hypersensitivity. Okay, so this was the initial part. So this you can find in Sharma, which is very well given over there, or you can read it from the microbiology book. Generally, the question is not asked on this part in pharmacology. We are only concerned about the drugs. Okay, so what are the types of drugs in immunopharmacology? They are immunosuppressants and immunostimulants. What are the major classes of immunosuppressants? The major classes of immunosuppressants, one group of drug you have already seen, that is glucocorticoids, okay, which are the inhibitors of lymphocyte gene expression, okay, to reduce the inflammatory response. The second group is the group of calcineurin inhibitors and mTOR inhibitors. mTOR is nothing but mammalian target of rapamycin. Okay, so inhibitors of this calcineurin or mTOR, which inhibit the signaling in the lymphocyte to prevent the cell activation. Then we have some drugs which you have seen in cancer chemotherapy, that is anti-proliferative and cytotoxic drugs like azathioprine, cyclophosphamide, okay, mycophenolate, mofetil, and we have biologic drugs or antibodies, mainly the anti-cytokine antibodies. Okay, anti-cytokine antibodies like the ones you have seen in treatment of rheumatoid arthritis, TNF alpha inhibitors, okay, interleukin 1 inhibitors, interleukin 2 inhibitors, and interleukin 6 inhibitors. There are so many which are given in your textbooks, but the main ones are tumor necrosis factor alpha inhibitors, interleukin 1, interleukin 2, and interleukin 6 inhibitors, which are also the monoclonal antibodies. Also, we have polyclonal antibodies. Okay, polyclonal antibodies like anti-thymocyte globulin and RHD immunoglobulin, which is used in uh, the hemolytic disease of the newborn when the female she has Rh negative blood group and the fetus it has Rh positive blood group. To prevent the immune reaction in the next pregnancy, we give this Rh D immunoglobulin, and that is an example of polyclonal. So how do we classify immunosuppressants? You can remember the classification which is given in Sharma. In Sharma, they have given the classification like this, and this classification is from Tripathi. I prefer this classification because it is easier. Okay, immunosuppressant drugs, they are classified into calcineurin inhibitors, mTOR inhibitors, anti-proliferative drugs, glucocorticoids, and biological agents. Okay, you will understand this calcineurin inhibitors and mTOR inhibitors when you see the mechanism of action of cyclosporin, tacrolimus, and sirolimus. Okay, so take a look at this picture. Over here is a macrophage, which is the antigen presenting cell, and the MHC2 class antigens which are present on the macrophage cells. Okay, they process the antigen peptide, and this antigen peptide is presented to the CD4 cell. This is a T helper cell or CD4 cell having the presence of T cell receptor. Okay, so when this antigen is presented to the T cell receptor, and also there is co-stimulation, that is CD28 stimulation by B7, that is CD80 and 86, any one of them which combines with this CD28, it will lead to the activation of this T cell receptor. When there is activation of this T cell receptor, there will be phosphorylation, formation of this inositol diphosphate, inositol triphosphate, and increase in the levels of calcium, which combine with calmodulin, and this calcium calmodulin complex, it attaches to this protein in the T cell, which is a calcineurin. Okay, so this is calcineurin, and calcineurin is activated by calcium calmodulin complex, right? So what this calcineurin it does, calcineurin it dephosphorylates NF80, okay, nuclear factor associated an NFAT is dephosphorylated, and this NFAT then will transduce inside the nucleus, and it will lead to the generation of cytokine, that is interleukin 2, and interleukin 2 is released, and again this interleukin 2 acts on the interleukin 2 receptor, and the main receptor over here is CD25, and that leads to the T cell proliferation. So here there is presence of mTOR, which is a mammalian, which is a mammalian type receptor of rapamycin, and this mTOR causes the stimulation of kinases, and the cell, it goes from G1 stage to S stage. Okay, that is leading to T cell proliferation. So this is the cycle of activation inside the T cell. Okay, the main role is played by two things. One is calcineurin over here, which dephosphorylates this NFAT, and second is mTOR. So cyclosporin, what is its action? Cyclosporin, it enters the cell and it attaches to a protein cyclophilin, and cyclosporin complex then inhibits the calcineurin. That is why cyclosporin is known as a calcineurin inhibitor. Okay, tacrolimus, it has same action, that is, it is a calcineurin inhibitor, but it attaches to another type of a immunophilin. The cyclophilin is a immunophilin. It attaches to another type of a immunophilin or an intermediary protein, that is FKB12. So tacrolimus combines to FKB12, and this combination inhibits calcineurin. That is why cyclosporin and tacrolimus, they are known as calcineurin inhibitors. Okay, sirolimus, it attaches to the FKB protein, FKBP protein, similar to tacrolimus, but the combination of these two inhibits mTOR, and that is why the sirolimus is known as mTOR inhibitor. Okay, so now you can understand this classification in a better way. Calcineurin inhibitors, they are cyclosporin and tacrolimus. mTOR inhibitors, it is sirolimus. You can remember sirolimus, a single drug over here. Anti-proliferative drugs, they prevent the proliferation of the T cells, that is azathioprine, methotrexate, cyclophosphamide, which is alkylating agent, and mycophenolate mofetil. Glucocorticoids, they have already been covered, and then we have biological agents like polyclonal antibodies, anti-thymocyte globulin, and Rho D immune globulin. Then we have TNF alpha inhibitors, interleukin 1 antagonist, interleukin 2 antagonist, and anti-CD3 antibody. Okay, where is the CD3 present?
In TCR.
Yes, in the T cell you have seen when this T cell, when this MHC2 presents over here with the peptide fragment, there is stimulation of CD3. Okay, so this CD3 is inhibited by muromonab, which is a very old antibody which was synthesized in 1980s, but still it is used. So this is the classification of immunosuppressants. Okay, I think you remember the TNF alpha. Yes.
I'm really sorry to disturb you, sir.
Okay.
Sir, can you please tell me the role of protein kinase C in the previous slide?
Protein kinase C is responsible for its role was here. Okay, protein kinase C induction, it leads to the switch of the immunoglobulin production from IgM to the other antibodies. Okay, you mean to say in the mechanism of action?
Yes, sir.
Okay, so this protein kinase C is also responsible for the activation. Okay, similar mechanism. But the main mechanism over here is
From
because of calcium, leading to formation of calcium calmodulin complex, which activates the calcineurin.
from where this FKBP protein came?
FKBP protein is present inside the cell only. Okay, it is present inside the T cell. In the T cell, there are various types of proteins. Okay, they are immunophilins which are already present in the T cell, like FKBP protein, FKBP12. Okay, and also this cyclophilin. So they are already present in the T cell. Okay, is it clear?
Yes, sir. Yes.
And they
Thank you so much, sir. Thank you so
they are
much.
the binding sites for the drug. FKBP12 is binding site for tacrolimus as well as sirolimus, whereas cyclophilin is the binding protein for cyclosporin. Okay.
Yes, sir. Then, sir, then what does this FKBP and sirolimus conjugate does?
Okay, FKBP and sirolimus complex, it inhibits the mTOR. Okay, mTOR plays an important role in production of cell dependent kinases. Okay, this cell dependent kinases, they cause the conversion of the cell cycle from G1 to S phase and play a role in cell cycle. They play a role in
Yes, sir. Thank you so much, sir. Thank you
cell
so much.
cycle proliferation. Okay, so when we inhibit this mTOR, the cell will be staying in the G1 phase only. Okay, and it will not proliferate. So sirolimus, when it combines with FKBP, it inhibits mTOR, and which will ultimately inhibit the switch from G1 to S phase, and the cell will be stuck in the G1 phase of the cell cycle. Okay, and this
Yes, sir. Thank you so
T
much,
cell
sir.
proliferation will be inhibited.
Yes, sir. Yes.
So you have seen the drugs for rheumatoid arthritis, that is TNF alpha inhibitors, right? Etanercept, infliximab, and adalimumab. Interleukin 1 receptor antagonist, that is anakinra. Interleukin 2 receptor antagonist, that is basiliximab and daclizumab, they are actually anti-CD25 antibodies. Okay, they are monoclonal antibodies which are targeted against the CD25, and they are basiliximab and daclizumab. Anti-CD3 is muromonab, and polyclonal antibodies, they are anti-thymocyte antibody and RHOD immune globulin. So now coming to cyclosporin. Okay, cyclosporin is a calcineurin inhibitor. What is its mechanism of action? Cyclosporin, it binds to cyclophilin to form a complex which inhibits calcineurin. Okay, so it binds to cyclophilin, and this calcineurin is inhibited. Okay, so dephosphorylation of the NF80 receptor, which helps the cytokine genes for production of interleukin 2, tumor necrosis factor, and interferon. So that will be inhibited. Pharmacokinetics of cyclosporin, it can be given orally as well as intravenously, but intravenously it is given very cautiously because it is nephrotoxic, and oral absorption is only 30%. Food decreases its absorption. What are indications of cyclosporin? Cyclosporin is indicated for prevention of graft rejection in solid organs. Solid organs like liver, kidney. Okay, it is also a second line drug in the autoimmune diseases like rheumatoid arthritis and psoriasis. It is not used as a first line drug because of its toxicity. Also, it can be used in other inflammatory diseases. Adverse effects of cyclosporin. Remember the H. Okay, hepatotoxic, hypertensive, hyperuricemia, hyperlipidemia, and hirsutism. Also, it is nephrotoxic and neurotoxic and causes gingival hyperplasia. Which is the other drug which causes gingival hyperplasia?
Phenytoin.
Phenytoin. Excellent. Phenytoin. So these are the adverse effects of cyclosporin. In the next class, we will see the remaining drugs. I think this is sufficient for today. The main important thing in the today's class is the classification. Okay, how the immunosuppressant drugs they are classified? They are classified as calcineurin inhibitors, mTOR inhibitors, anti-proliferative or cytotoxic drugs, right? Then the glucocorticoids and the biological drugs. In biological drugs, we have TNF alpha inhibitors like what are the examples? Etanercept, right? Any other example of TNF alpha inhibitor? Infliximab and adalimumab. Okay. Interleukin 1 inhibitor, that is anakinra. Interleukin 2 inhibitor basiliximab and daclizumab. Anti-CD3, that is muromonab, and polyclonal antibodies, which are two, that is anti-thymocyte globulin and Rho-D immune globulin. Any questions? Any questions or queries? So generally a short note is asked on this topic. Okay, you may be asked a short note on any one of the immunosuppressant like cyclosporin, tacrolimus, or sirolimus, or the other drugs they are covered elsewhere, right? Except for anti-thymocyte globulin and Rho-D immunoglobulin. I think all others they are covered somewhere or the other. Like azathioprine, it is covered in cancer chemotherapy. Okay. Also cyclophosphamide and methotrexate. Glucocorticoids, already you had a lecture on glucocorticoids. Okay. So from this topic, the main things are the calcineurin inhibitors and mTOR inhibitors. Any other question? If there is no question, thank you very much. And stay safe. Still, corona is not over. Day by day, the cases they are increasing. Okay, so stay at home, stay safe. Thank you very much.
Thank you so much, sir. Thank you so much, sir.